Hi everyone. Hope you guys had a good week. I'm gonna give people just one more second if some people are still joining. Give people one more minute before we get started. We're reaching the end of this course. Today we are going to cover, or last week we looked at genetics. We started genetics and we're going to do a small review over what we looked at last week. And then finish off genetics talking about pedigrees, which is just how genetics can be represented through generations. And then we'll move into DNA replication and translation. DNA is what holds all of your genetic information. What makes you you, essentially, and we constantly need to be reproducing or replicating DNA. As we grow, so DNA replication, transcription, and translation are the processes of how that happens and how DNA just duplicates and makes more, and how that's essential to our bodies. Sorry, it's raining outside. Today I don't have a Blue Kit plan, but I think what I'm going to do today is, or after we cover a little bit about genetics, we'll go probably through DNA replication and transcription. Next week we'll do DNA translation and then end with a just review. We can do a review and a Blue Kit next week on DNA, and then that'll be our final class. So yeah. Okay, I'm gonna go ahead and just get started and share my screen. You guys hear that thunder? I don't. There you go. All right. So okay, last week, let's take a look at what we looked at last week really quickly. So Gregor Mendel is the father of genetics, is what he's tokened as. And he essentially found that we have these things called alleles, which can be dominant or recessive. You guys remember that? And these alleles, they can code for specific genotypes, which can be represented in a person as a phenotype. So your phenotype are going to be how something physically appears. So for instance, I have black hair. That is a phenotype. But the genotype for black hair will be a code of alleles, something like this. So you have a couple different combinations of genotypes. You can have either a heterozygous genotype or a homozygous genotype, and that can be either dominant or recessive. So your heterozygous ones, again, hetero, the prefix means different, and different, that can help you understand that the alleles will be, they won't be the same, they'll be different. So one will be a capital uppercase letter and one will be a lowercase letter. And then what does the capital letter mean? That means it's a dominant allele or dominant trait, which will cover or mask the recessive one, the little h. Then we can also have homozygous genotypes, which are homo, that prefix means same or both same. And so that can tell you that we have two uppercase letters or two lowercase letters. The uppercase ones, again, are dominant, and the lowercase ones are recessive. Um, and then we saw how we can figure out the probability of these genotypes using a Punnett square. And this Punnett square can tell you, especially if you're looking at sex-linked sex-linked, what's it called? Disease or traits. Punnett squares are really helpful because they can tell you the probability of a certain offspring having that trait. And one thing I do want to point out again is that the females have their they have chromosomes X and X, and males have chromosome X and Y. And when we want to look at and when we want to analyze if a trait can be passed down to offspring, these traits are these recessive allele traits will typically only be represented on the X chromosome, meaning because the male only has one X chromosome, they have a higher probability of representing this trait, of being affected by this trait. Whereas in a female, if they have a dominant H here and they have a lowercase h, the dominant one is going to cover that recessive lowercase h. So they will be a carrier, but they won't be affected by whatever this trait is. And this trait could easily be some kind of illness or disease that is genetic. Whereas if the father had that trait, had a little h, they can only have one h. And so they will represent that trait. All right, now we're going to move on to pedigrees. So pedigrees, this will look, okay. Pedigrees show how a trait or disorder is passed down from one generation to the next. So it's really just a graph. And we can and it's really all about how you can interpret and read a pedigree. So let's take a look at how we can interpret this. Okay. So interpreting a pedigree, if it skips a generation, it's recessive. But if every generation has the trait or disorder, then it's a dominant disorder or dominant trait. So let's look at this pedigree on the left here. So the squares represent males and the circles represent females. The colored in shapes show who is affected, who has the disorder or has the trait. So here we start with, so this pedigree represents 3 generations. This could be the grandfather, grandmother. They have 3 kids, 2 males and 1 female. And then this male marries another female. This male marries another female. This female has no partner. So this pair of grandparents, the female is affected. The female has this trait or has this disorder. That's why it's colored in purple. Now, when they have these 3 kids, one of the males is going to be a carrier of this trait. Another one of the males will be affected by this trait. And then the female will not have this trait. Now, when this male marries this female and they have a kid, which is another female, this female will be an affected individual. So, and then this male represents a carrier because it's only half of the square is colored. All right. So what can we read or what can we learn from this pedigree? Because every single generation there is somebody affected. You have this female here, this grandmother, this other male here, and then this other female here, because somebody in every single generation is effective or affected, we know that this is a dominant trait. If it affects women and men equally, it is an autosomal trait or autosomal disorder. And what does autosomal mean? Autosomal means body cells. So at the very beginning or near the very beginning of this biology class, we looked at autosomal um, versus sex linked. And autosomal are your body cells. So those are like your skin cells, your hair cells, um, your nails, your organs. Those all have cells, and those are autosomal, your body cells. Now, your sex linked ones are your gametes. Um, and that's what we looked at like this. This is a sex linked trait or sex linked uh, Punnett square because you have this trait on your X and X chromosomes and your X and Y chromosomes, which are your sex chromosomes. Whereas in this other Punnett square, these would be autosomal. These code for autosomal because you don't have those X and Y chromosomes. Okay, so when it affects women and men equally, this you know that it's an autosomal trait. So it is a trait being expressed on like body cells. But if it affects men more than women, you have a sex-linked one. So what do we think here? Do we think that this is an autosomal trait and disorder, or do we think that this is a sex-linked one? You have two females and basically one male being affected. So are there more men being affected here, or are there more women being affected here by this trait? Yeah, more women are being affected here. Um, this means that it's an autosomal disorder or autosomal trait that we are reading. Females tend to carry a trait. If females tend, and here are some patterns, sorry, here are some patterns that we can just learn in general from pedigrees. If females tend to carry a trait, they then it'll typically affect their sons. So here this female carries a trait and they have two sons. Both sons essentially are affected. The females usually get a trait from their affected father or affected mother. So here you have this female who is affected and she got it from her affected father. But affected males got it usually get it from their mother and then give it to their daughter. So we see that pattern here. Okay, let's look at another pedigree. So here we start. Okay, how many generations are we looking at here? Here we're looking at four generations. So we start with this pair, um, and you have an affected father. They have a lot of children. They have, how many is this? They have 5 children. Two males and 3 females. All 3 females are affected, but none of the males are affected. All right. And then they have a bunch of kids, they marry. Um, then we see some of the trait, then we see the trait being expressed in some of the males, um, also some of the females. And then in the next generation, we mostly see females again. So, using all this information, is this a dominant or recessive trait? Do we see this in every single generation? Does it skip any generations? Yeah. So, because we see this in every single generation, we know that this is a dominant trait. And now, do we think that this is autosomal or sex-linked? Are the women and men affected basically equally, or are there more men being affected? Yeah, nice. Good job. So yeah, essentially more women are being affected here. There isn't a large amount more men. It's pretty even. So we know that this is an autosomal trait. All right. And now I'm going to introduce DNA. After I finish the slide, we're going to watch a video introducing DNA, and then I'll go further in depth with DNA replication and DNA transcription. And that'll be that for today. Next week, we'll go into translation. Okay, so this is your DNA. DNA is, you usually refer to or you usually see DNA being referred to as a double-stranded helix. So this is what we see here. It has 2 strands and it forms this sort of helix here, curvy helix. And there are 4 nucleotides. So do you guys remember our macromolecules? What were our macromolecules? We had carbs, fats, proteins, and nucleic acids. So your nucleic acids are what we're looking at here. The monomers for nucleic acids, which make up our DNA, are these nucleotides. And we have 4 nucleotides, or 4 nucleotides in DNA, which again hold our genetic information. They won't make us ourselves, and why we're different from our parents. So we have adenine, thymine, guanine, and cytosine. ATGC. Adenine, thymine, guanine, and cytosine. Now, sorry. Then we have the DNA model. The DNA model was made by Watson and Crick, and they basically found that the nucleotides, specific nucleotides, will pair up. So they found that adenine, or A, always binds with thymine, and guanine always binds with cytosine. So A binds with T and G binds with C. So if we look at the DNA strand on the right, the G is going to bind with the C, A binds with T. And you'll see that all throughout DNA. And this is what's called the DNA model. All right. Now we're going to watch a video introducing DNA replication. Okay, I like this one.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors? In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. Okay, so I hope that provided just like a better overall understanding of what we're going to be learning. So now we're going to start a little bit more in depth with DNA replication. Give me 1 second. Sorry, I had to move to a desk. Okay. So as the Amoeba Sister says, we start with this first strand of DNA. Helicase here, sorry. This is going to be, okay, so here's your leading strand of DNA. This is your lagging strand. So it's going to go this way. And then you have your topoisomerase, which is unwinding this helix here. So first you have your strand of DNA. Helicase unwinds the double helix from forming helicase unwinds the double helix, which forms the replication fork. And this is the replication fork right here. It's sort of like when it splits off into two ends. So you have your helicase separating and unwinding this double helix here. Topoisomerase prevents the DNA strand from supercoiling. So, trying to think of a good example. Okay, so have you guys ever had like played with a slinky before? I don't know if you guys have slinkies. Too old. But slinkies or a spring, really. You unwind the spring, you stretch the spring out, there's going to be that force that wants to pull it back in, and that's what topoisomerase essentially stops. It holds that slinky in place, so when you unwind it, it won't like want to supercoil and retract back. So that's what topoisomerase does. It holds it in place. And then the proteins stabilize the exposed single-stranded DNA. Oops, sorry. So these proteins are just stabilizing this exposed strand of DNA. Then we have RNA primase, which primes the strand of DNA. So basically, this primer or DNA primase, both essentially are doing the same thing. They prime the strand of DNA before more DNA attaches to it. And then at the replication fork, enzyme poly polymerase, DNA polymerase here moves along each strand and adds the complementary nucleotide. So remember that A binds with T and G binds with C. Adenine binds with thymine and guanine binds with cytosine. So they're adding the complementary ones to each strand of DNA after this DNA DNA primase primes each of the exposed strands. And then this continuously happens to the leading strand as it's synthesized from 5 prime to 3 from sorry, 5 prime to 3 prime. So here's your leading strand. It's being unwind, but this top strand is your 5 prime, 5 prime to 3 prime, so it's being synthesized this way first with the leading strand. So here's some terminology that you guys will probably want to remember if you're going to take a biology class in the future. DNA replication runs anti-parallel, so it goes in opposite directions. You'll also hear semi-conservative replication, where the original DNA strand is the template for the new DNA strand. But because you have that original DNA strand that split, and then you add onto like the other half or the complementary DNA nucleotides, you have a semi-conservative replication. So you still have that original DNA strand, but now half of it is new. And again, it replicates in the 5 prime to 3 prime direction. Now we're looking at the lagging strand. So here at the replication fork, your DNA polymerase, which again is an enzyme, which because it ends with ASE, things that end with ASE, or typically enzymes, they move along each strand, adding complementary nucleotides this way. But because the polymerase can only synthesize in the 5' to 3' direction, it constantly needs to catch up as the helix unwinds, which is sort of difficult to visualize, but essentially it's going this way. This is unwinding. This is unwinding, so it constantly needs to just like keep it up and pick up, and keep working. And because it's working in 5' to 3' direction and it constantly needs to keep up with the helicase, it's going to create these sort of Okazaki fragments. Okazaki because that's the person who discovered these fragments on the lagging strand. So here, on the lagging strand, which is this top one, we have DNA polymerase. The helicase is going to unwind this way to the right, but DNA polymerase can only go from 5' to 3'. So it's going like this way to the left. So it's adding complementary nucleotides, then it's going forward and doing it again, and going forward and doing it again as this continuously unwinds. So it's like jumping, going, moving forward, and then going again. And that's what's creating these fragments on the lagging strand. It's a good thing, though, we have DNA ligase, which glues the fragments together. So they're not just left as fragments. Your ligase is going to bind these fragments together so it's one big strand again, which is very interesting because we have all of these enzymes and all of these little pieces inside of us to make sure things run smoothly. It's a big, complicated system, but it ensures that we function properly and are healthy and live. Now for transcription, which is when the DNA sequence that we had just replicated is transcribed into mRNA. And mRNA is just the step in between before we that helps code for full proteins. So mRNA is essentially the code for the proteins that we want in translation. We're translating this mRNA from the original DNA, and then we're translating this RNA into proteins, which is what we want in the end. So, or amino acids, which are the same thing. So it's initiated when an RNA polymerase, so very similar to DNA polymerase, binds to the promoter binding site. So there is just a promoter binding site on this new strand of DNA, and they'll just bind to that. That is just the signal that it gets, or the sign that it gets, like, hey, I'm the promoter binding site. This is where you bind, and that's where it starts. Then it moves along the DNA strand and adds corresponding complementary nucleotides. So, one difference between RNA and DNA is that RNA has no thymine, which binds with A, adenine. Again, A binds with T in DNA. But because RNA has no thymine, it's replaced by something called uracil, which is another nucleotide. And so instead of A binding with T, A binds with U, which you'll see here. Okay, so like DNA polymerase, which adds the corresponding nucleotides, RNA polymerase is going to unwind the DNA that we just duplicated or replicated and do the same thing. So it moves along the DNA strand and adds corresponding nucleotides until it receives a stop signal. One side of this DNA is going to code for protein, and that's where the mRNA is transcribed from the DNA. So you have this mRNA here. At the image to the right, the RNA polymerase splits up this DNA that we just duplicated, and is going to find pairs to each of these nucleotides. So A binds with U, and this was the original T, which is why A binds with T, but that's going to split up and become U, and then GC, GC, AU. It's like a puzzle, essentially. It's splitting that DNA up and it's looking at all of those nucleotides, and then the RNA polymerase is going to transcribe all of these on the bottom and create another strand called mRNA. And this essentially transcription bubble moves down DNA at a constant rate, and this is called elongation. So your mRNA strand is being elongated constantly. Oh, sorry, it's going this way, not to the left. It's going to the right because it's from 5' to 3'. And then the non-transcribed strand is called the codon strand. So when RNA polymerase splits up this DNA and opens it up like that, only one side is being transcribed this way. And then the other one that's not being touched is called the codon strand. And then afterwards, um, the RNA polymerase is going to receive the stop signal. It's going to detach, and the DNA will go back to their original pairs. It's just going to close up. Okay, that is where I'm going to stop today. Next week, we're going to finish off with translation and do some blue kits and just overall review, and that'll be our last class. So we're reaching the end of this course. I hope you guys have been enjoying it so far and are learning something. But yes, I'll see you guys next week. I think pretty early today. Bye. Have a good week.